everyone. Hello everyone. This is the next video lecture on sexual reproduction in flowering plants. And the last video lecture in this topic we mainly studied about the pollen pistil interaction. I think students all of you understood about pollen pistil interaction. The important one, please remember that, and how the pollen is going to be selected by pistil or the stigma. Students, you know already there is a series of events are going to be taken place between the pollen grain and the stigma. Usually stigma is going to be select a right type of pollen, and it is going to be reject a wrong type of pollen. Either it is self incompatible or it is from the other species, or the pollen grain is from the other species. That we already studied, and in that only how the pollen grain is going to be start development. There is a germination occurs when the pollen grain come and settle on the stigma. Germination occurs, and germination leads to that there is a development of the pollen tube that will run towards the ovary, and the fertilization occurs over there. Okay, that complete scenario we came to know about from the pollen pistil interaction. And now coming to the next one, that is artificial hybridization. Okay, that is artificial hybridization. First we should know about the meaning of word hybridization. It is nothing but the production of hybrids. Don't go through chemistry definition, just remember in biology how it is. The hybridization is nothing but the production of hybrids. Say for example, we have plant A, we have plant B, and we have plant C. Some desirable characters are present in A, and some desirable characters are present in B species. And I don't want C because it do not contain any desirable character. Okay, it do not contain any desirable. I have to cross these two plants. Okay, how to transfer the pollen grain of A to the B to the A plant. Okay, because I want these characters, B plant characters. So I have to transfer the pollen grains from B to A. Either you can transfer from A to B. We have to select the particular characters, or we have to select the particular desirable pollen grains. We need that because they do not contain any desirable characters. Why should we use the pollen grain of that? We need a variety, we need a product which is different from these two. For that purpose we are going to be produce the hybrids. Okay, we are going to be produce the hybrids by using two desirable characters from different plants. Okay, by doing that we can able to produce the hybrids. How we are going to do it? It is we are going to be do with the help of with artificial method. Okay, so it is the artificial hybridization. It's nothing but artificial hybridization. Okay, simple definition, you can remember that. For what purpose it is? Just to improve the crop yield. Just to improve the crop yield. We can do in case of animals also. This we can do in case of animals also. And in case of plant also we need this. Because within a short period we need more amount of food. For that we are completely depends on the hybrid. We already depend on the hybrid, because if you go through the normal natural methods, it is not possible to get the more amount of yield in a short time. But with the help of hybrids we can get more amount of yields in a short time. Okay, just to improve the crop yield purpose we are using hybridization. Okay, and here in case of hybridization we are going to be we are going to be select the plant. First we have to select the plant, and after that, after the selection, what we are going to do? Okay, first what we have to do, we have to select the plant. Okay, what type of characters we want and what characters we need and we will select first the plant. Okay, from which plant we want pollen grains, that plant we have to first select. That is the first selection is very important. And this hybridization is mainly achieved by two methods. Okay, not two methods, they are going to be achieved by two important aspect or two important concept. One is emasculation, another one is bagging. Okay, one is emasculation and bagging. And here students, what purpose we are using these two? Because we have to collect the pollen grains. Because naturally what happen, pollen grains are going to be continuously transferring from one plant to another plant, or it may it may there is a self pollination also, but we have to avoid that. And along with that we have to protect the stigma from unwanted pollen grains, because naturally what will happen, so many different types of pollen grains from the same plant or from the different plant, naturally it will happen. But what we need desirable pollen grains, not unwanted pollen grains. For that we have to protect the stigma from unwanted pollen grains, and we have to ensure the right kind of pollen grains. Okay, we have to ensure the right kind of pollen grains from the right plant, and these two are mainly achieved by emasculation and another one is bagging. Emasculation and another one is bagging. Usually students, I will show you the another slide. Okay, usually students, the plants will produce bisexual flower. Means both the sexes are present in a same plant. Hermaphroditic flowers are going to be produced here. Hermaphroditic flowers are going to be produced here. Both male and female reproductive systems are present here. So what will happen if the both, this is the gynoecium and these are the androecium. This is gynoecium and androecium. Okay, gynoecium and androecium. What will happen here? The pollen grains are able to transfer from here to here. Okay, but we don't want these pollen grains. We don't want these pollen grains. We want the pollen grains from another plant which contains desirable characteristics. Which contains some desirable characteristics. These pollen grains we have to transfer from this plant to this plant. That we needed. Okay, for that purpose we have to follow two important steps or two important methods. One is emasculation, another one is bagging. Okay, emasculation and bagging. Okay, listen, first here what will happen? First step, that is emasculation. Okay, say for example this is the stigma here, this is the stigma here, this is the androecium here, you can find androecium. Okay, and first, in case of emasculation, please remember students, this emasculation is going to be asked for your exam as a 1 marks question, or sometimes they will ask for 2 marks also. For that purpose you have to remember the definition of emasculation. Here we are going to be remove the anthers. We are going to be remove the anthers from the flower. This is the emasculation, the process of removal of these anthers from the flower, because we don't want these pollen grains. Okay, we don't want these pollen grains because we are not finding any desirable character from this anther. Okay, just for that purpose we don't want these pollen grains, so we have to remove the anthers. And the process of removal of the anther from the flower, that we call it as a emasculation. That process we call it as a emasculation. You can see here, that process we call it as a emasculation. Okay, here the anthers are going to be removed from the buds. Okay, and that method or that process we call it as a emasculation. Please remember the process of removal of anthers from the stigma. The process of removal of anthers from the stigma, sorry, not from the stigma, removal of the anthers from the flower. Okay, that method we call it as a emasculation. Okay, once we did the emasculation, okay, once we completed the emasculation, after that there is a bagging. Okay, after completing the emasculation, we'll go through bagging. And in bagging, what will happen? First, here this is the female reproductive part, that is stigma. Complete gynoecium is here. Okay, here is the ovary, and then inside this there you can find the ovule. And here, the after removing the anthers, for example, now I removed the anthers. Okay, these do not contain any anthers. Just will go for bagging. Just will cover the stigma with the help of bags. That method we call it as a bagging. For what purpose? Just to avoid the unwanted pollen grains. Just to avoid the unwanted pollen grains, those are transferring from different plant or from the different species, or just from the same flower, self pollination. Okay, for that purpose we have to cover the stigma with the help of bags. With the help of bags, this method we call it as a bagging. And after bagging, here before that, when the stigma is ready to receive, now the stigma is able to receive the pollen grain. At that time, what we are going to do, just will remove the bag from the stigma, and we will add, remove the stigma from, sorry, bag from the stigma, and we will add the pollen grains. Okay, or we will add the desirable pollen grains from the different flower. We'll add the desirable pollen grains from the flower. Okay, from which flower or from which plant we wanted to collect the pollen grains, those are going to be collect and then transfer these to the stigma. Then we will get the product, or we will get the hybrid. Okay, hence we will get the hybrid. This is in case of bisexual flowers, in which their plants are producing both male and female sexes in the same plant. In such condition, we need that. Okay, and after that, here in case of unisexual flowers, we will not do this method. Okay, just here the same thing, just will not remove the anthers, because there is no anthers. Okay, in case of female plant, there is no anthers, because that female plant producing only female flowers, and male plant producing the male flowers, so no need to remove the anthers. Okay, just we will undergo for bagging, just to avoid the unwanted pollen grains, those are coming from the different flowers. Okay, those are coming from the different flowers. Okay, when the stigma is receptive here, okay, in case of unisexual, when the stigma is receptive, at that time we'll remove the bag and we'll transfer the pollen grains. Okay, we'll transfer the pollen grains. Again we are going to add the bags. Add the bags, and we will allow for the further development. Okay, we'll allow for the fertilization, and then there is the development of the embryo. Okay, seed is going to be developed, and that for that purpose we have to allow them. Okay, this is the hybridization technique what they given in your NCRT. Okay, and here some points I have given here. Here bagging and everything I was given here, that emasculation and bagging. And here emasculated flower is covered by a bag. Okay, bag, we are going to be cover the flowers or emasculated flowers with the help of bag, just to avoid the unwanted pollen grains. Okay, and after completing the pollination, again we will go for rebagging. Okay, again we will add the bags, and that is we call it as the rebagged condition. And then finally there is a development and development of the embryo, and then seed is going to be developed. Okay, this is the artificial hybridization, the method of artificial hybridization. Okay students, I think all of you understood about the hybridization, artificial hybridization. And in this concept exactly we need definition of emasculation, okay, and bagging. Please remember these two definitions because these are important for your exam point of view. Okay, these are important, little important. Please remember all those. Okay. Okay, after completing the artificial hybridization in your NCRT they given double fertilization. Okay, they given double fertilization. What is meant by double fertilization? Or before that we have to know about the fertilization. Fertilization commonly we know that that is fusion of the male and female gametes. Okay, fusion of the male and female gametes or fusion of the gametes we call it as a fertilization, and or it is also called as syngamy. It is also sorry, it is also called as syngamy. It is also called as syngamy, fusion of the gametes, that we call it as a syngamy. Here during fertilization what will happen? Double fertilization. Usually in case of flowering plants there is a double fertilization. Fertilization is two time or fusion of the gametes is twice. Two time fusion of the gametes occurs here. Okay, for that we call it as a double fertilization. I will show one video on this concept later. First I will explain the things. Okay, first what will happen here? I will take one empty slide. Okay, okay, this is empty slide. Here you can see uh, first I will draw the female reproductive system, that is gynoecium. This is the gynoecium. Okay, this is the gynoecium. Here there is a presence of ovary, and inside the ovary you can find here ovule. Okay, inside the ovary you can find here ovule, and inside the ovule here it contains the embryo sac. Embryo sac is located here, and it is mainly contain different types of cells. Okay, that will later we'll discuss about that. What will happen here when the anthers when the anthers release the pollen grains? When the anthers are going to be release the pollen grains, okay, and these pollen grains will settle on the stigma. They will settle on the stigma and they start germination. They start germination, and that will continue into pollen tube. That will continue into pollen tube. Okay, where from where it is going to be germinate? It is from the generative cell. Usually germ pores are there. Through the germ pores pollen tube is going to be emerged out from the pollen grains, and this here they are going to be developed here. This is the pollen tube you can see here, and this will run along the style and towards the microfilar end of the female gametophyte. Okay, microfilar end of the female gametophyte. If I draw the female gametophyte here, here you can see the structure. Okay, like this is the structure. This is the chalazal end, sorry, this is the chalazal end, and this is the microfilar end, and here you can see the integuments. Okay, this is the structure. These are integument, chalazal end, microfill, and here embryo sac, and here and usually the pollen tube will come and pass through the microfilar end. Okay, this happen in case of double fertilization. And here when you come to the exact structure of the embryo sac, okay, exact structure of the embryo sac, see here I will draw the diagram first here. This is the embryo sac. Sorry, wait. Okay, you can see this is the embryo sac, and in this embryo sac there is the presence of different types of cells we can see here. This is the chalagenate. Here you can find antipodal cells. 3 antipodal cells are present over there, and in the microfilar end we will find 2 synergid cells and 1 egg cell. And in the middle, just wait, 2 synergid cell and 1 egg cell is going to be present here. And in the middle there is a presence of polar nuclei. Here you can find the polar nuclei. These are the cells here, polar nuclei. These are the antipodal cells. These are the antipodal cells. These are the polar nuclei. These are the synergid cells, and this is the egg cell, which is present inside the ovary. Okay, when the fertilization occurs, so not fertilization, when the pollination occurs, the pollen grains are going to be develop their pollen tube towards the micropillar, and like this there is a traveling of the pollen tube towards the micropillar end. And in the micropillar end there is a presence of filiform apparatus. Here you can find the filiform apparatus, and these filiform apparatus will guide the pollen tube to reach the synergid cells. Okay, they will guide the pollen tube just to reach the synergid cells, and this pollen tube will carry 2 gametes. 1, 2. It mainly carries 2 gametes. When the piliform apparatus receives the pollen tube, they will guide the pollen tube to reach the synergid cells. When they reach the synergid cells, at that time what will happen? One of the gamete, I will draw here the diagram. Wait, you can see the diagram. Okay, and here these are the antipodal cells, and this is the egg cell. You can see here, this is your microfilar end. Here you can find the piliform apparatus. When pollen tube reaches here, it mainly contains 2 gametes, and piliform apparatus guide the pollen tube and it releases the gametes, and these gametes will reaches to the synergid cell. This reaches to the synergid cell, and synergid cell will guide the pollen tube towards the egg cell. Okay, it will give the guidance for the fertilization. Synergid cell will give the guidance for the fertilization, and one of the gamete is going to be fused with egg cell. Okay, it mainly going to be fused with egg cell. This is the first fertilization occurring here. First fertilization we can see here. First fertilization occurred here. 2 gametes are there. This is one. 2 gametes are there. Among these 2 gametes, one is going to be fused with the egg cell. And another 1 gamete is going to be fused with this polar nuclei. Okay, it's going to be fused with this polar nuclei. Already one is fused with here. Okay, and another one is going to be fused with the polar nuclei. Okay, first fertilization occurred in case of egg cell, and the later one with the polar nuclei. 2 times there is a fertilization occurs, so this we call it as a double fertilization. Twice fertilization is going to be happen, so we call it as a double fertilization. Okay, we call it as a double fertilization. Okay students, it is very simple. Double fertilization, please remember, important. Okay, it is very important. Pollen grains, when they settle on the stigma, they start germination, and that germination becomes a pollen tube is going to be formed, and that will run along the female gametophyte and reaches to the reaches to the ovary in the microfilament, and then it enters into the synergid cell. With the help of filiform apparatus, the gametes will enter into the synergid cell, and these synergid cell will guide the pollen, that gamete fuse with the egg cell. It becomes, when it fuses with the egg cell, it becomes 2n diploid, and another one is going to be fused with polar nuclei, that is become 3n. Okay, triploid. Please remember this one. They become triploid, and this egg cell is diploid. And whatever the polar nuclei is going to be fused with egg cell, it becomes 3n, and this we call it as a primary endosperm nucleus. Okay, primary endosperm nucleus, please remember this one. And further it becomes endosperm, and everything we'll study later on in post fertilization event. We are going to be study about that, but in the beginning, please remember this one. Primary endosperm nucleus is going to be formed, and when the fertilization occurs, the synergid cells, the antipodal cells, they are going to be disappear. Both synergid and antipodal cells, both of them are going to be disappear. Okay, they are going to be disappear, and further development, embryo and everything is going to be developed in that. Okay, this is going to be happen here. In your NCRT, they given one diagram. Here they given one diagram. Wait, I'll show that. Okay, in your NCRT, they given diagram. This is the diagrammatic representation, and students, please remember, this is fertilized embryo sac. This question asked so many times. This is fertilized, don't get confused. Okay, this is a fertilized embryo sac. Okay, this is fertilized embryo sac, please remember this. And here you can see degenerating synergid cells. Here these synergid cells are going to be degenerate here. These two synergid cells are going to be degenerate here. There is a formation of the zygote, which is 2n. Okay, because fusion occurred here, so there is a formation of zygote, that is 2n diploid. And in the middle here, primary endosperm cell, complete this cell we call it as a primary endosperm cell. This complete cell we call it as a primary endosperm cell, and that is PEC, that is PEC. And next to that, here you can see the primary endosperm nucleus, that is triploid, 3n. Okay, triploid 3n structure we can see here. And here the degenerating antipodal cells and synergid cells are going to be disappear. What remained here? Only the zygote, the primary endosperm cell, and the primary endosperm nucleus is going to be remain here, and the remaining all are going to be degenerate. And the further development occurs and the endosperm and everything, that we will study in the next video, that is in post fertilization events. Okay, this is about the double fertilization. Students, very simple concept. Please remember what will happen, what type of cells are there, and how the fertilization is going to be happen. Okay, very simple method, you can remember. Okay, there is no complication at all, very simple. In your NCRT also they given very simple and easy way. Okay, in a simple and easy way they given the double fertilization. Just you have to remember and remember the things what exactly going to be happen. Okay, this is about the double fertilization. This is about the double fertilization, and I think so all of you understood about this concept. If you have any doubts or queries regarding double fertilization and artificial hybridization, you can ask the question, okay, through WhatsApp. You can do the WhatsApp and you can ask the question. And these are all the points you can see here. These are all the points regarding double fertilization. This much is enough. Okay, and artificial hybridization also there is a points, you can follow these, and along with that you go through your NCERT textbook. Okay, this is the thing about the double fertilization and artificial hybridization. The next video lecture will go through post fertilization events. Okay, and post fertilization events we will discuss in the next video lecture. Okay, thank you students.